Welcome to the Learning Module for Group 2 hormone signaling involving receptor tyrosine kinases and other cell surface receptors. This is Dr. Tefke for the Cellular and Molecular Medicines course. After studying this learning module, students should be able to describe the receptor tyrosine kinase mechanism, explain the role of adapter proteins in signal transduction, describe the function of GRB2, SOS, and Ras proteins, explain how GEFs and GAPs control Ras activity, illustrate the MAP kinase cascade, explain how insulin receptor is different from other receptor tyrosine kinases, describe the function of the insulin receptor substrate family of proteins, explain the PI3 kinase PKB pathway mechanism, describe TGF-beta signaling mechanism, explain how receptors with guanylate cyclase activity produce their effects and provide examples, describe the activation of the JAK-STAT pathway by cytokine receptors, explain how cell signaling is integrated and terminated. Receptor tyrosine kinases is a relatively large family of cell surface receptor proteins that bind to polypeptides and proteins that function as growth factors. Receptor tyrosine kinases have an extracellular ligand binding domain, a single transmembrane spanning domain, and an intracellular cytosolic domain that has the catalytic activity of a tyrosine kinase. With very few exceptions, all receptor tyrosine kinases function as dimers. Ligand binding to the extracellular ligand binding domain of a receptor tyrosine kinase induces receptor dimerization. This places the cytosolic tyrosine kinase domains in close proximity to one another. They are activated and autophosphorylate tyrosine residues in their cytosolic domains. This creates phosphotyrosine residues, which are binding sites for a class of proteins known as adapter proteins. These adapter proteins have phosphotyrosine binding domains called SH2 domains. These domains specifically recognize phosphotyrosines on receptor tyrosine kinases. Binding or docking of an SH2 containing adapter protein to the receptor tyrosine kinase phosphotyrosines initiates formation of a multi-component signal transducing complex that ultimately results in the cellular response. Polypeptide proteins, usually larger than 50 amino acids, that function as growth factors to stimulate cell proliferation usually activate receptor tyrosine kinases. If the abbreviation for the hormone or growth factor has GF in it, it usually functions through a receptor tyrosine kinase mechanism. For example, NGF or nerve growth factor, platelet-derived growth factor, epidermal growth factor, fibroblast growth factor, vascular endothelial growth factor, etc., all use receptor tyrosine kinase mechanisms. There is one notable exception, the transforming growth factor beta superfamily, which we will be discussing later in this learning module. Adapter proteins function in the formation of multi-subunit relay complexes. Each adapter protein contains interaction domains that allow them to specifically form complexes with corresponding docking domains on other molecules. Essentially, the adapter proteins and the proteins that they interact with fit together like Lego pieces. There are four main classes of interaction domains found on adapter proteins. There is the phosphotyrosine binding domain, or the PTB, which recognizes phosphotyrosines, the Sarcomology domain 2, or SH2 domain, which binds to specific phosphotyrosines, the Sarcomology domain 3, or SH3 domain, which binds to proline-rich sequences, and the plextrin homology or pH domain, which binds to phosphoinositides in the inner leaflet of the plasma membrane. Growth factor binding of a receptor tyrosine kinase will activate the mitogen-activated protein kinase signaling pathway. This involves two adapter proteins known as GRB2 and SOS, as well as a guanine nucleotide protein called Ras. GRB2, which stands for growth factor receptor bound protein 2, has an SH2 domain for docking with the phosphotyrosines of an activated receptor tyrosine kinase. SOS, which stands for son of sevenless, has an SH3 domain for docking with the proline-rich domain of GRB2, which becomes exposed when GRB2 binds to the activated receptor tyrosine kinase. SOS then activates a guanine nucleotide binding protein called Ras. By promoting the exchange of GTP for GDP, Ras is activated. Ras proteins are a family of small monomeric guanine nucleotide binding proteins that function similar to the alpha subunit of heterotrimeric G proteins involved in G protein coupled receptor signaling pathways. Ras bound to GDP is inactive. Ras bound to GTP is active. Activated Ras can then bind to and stimulate the activity of a downstream kinase known as mitogen activated protein kinase, or MAP kinase 3, sometimes called RAF. Some forms of Ras are post-translationally modified with a prenyl group. This prenyl group is hydrophobic and inserts into the plasma membrane, thus anchoring Ras at the inner plasma membrane surface in close proximity to receptor tyrosine kinases. Since prenylation of Ras is necessary in some cases for mitogenic signaling, inhibitors of the enzymes that prenylate Ras are under clinical investigation for use in cancer therapies. Adapter proteins, like SOS, that function to promote the exchange of GTP for GDP on Ras and thus activate it are what are known as guanine nucleotide exchange factors, or GEFs. GEFs function to activate Ras. Similar to the alpha subunits of heterotrimeric G proteins, Ras also contains an intrinsic GTPase activity, which functions as an auto-timer mechanism to limit the amount of time that Ras is in its active form. GTPase activating proteins, or GAPs, are a family of protein that function to accelerate the GTPase activity of Ras and thus shorten the period of time for which Ras is in its active GTP-bound form. Neurofibromin, or NF1, is one member of the GTPase activating proteins which function to promote the GTPase activity of Ras to inactivate Ras signaling. Loss of function mutations in the NF1 gene lead to neurofibromatosis. The NF1 protein has a deficiency in GAP function. This leads to sustained Ras-GTP stimulation in the tissues along the nerves, causing them to grow uncontrollably and form nodules, called neurofibromas. The individual shown in this picture has an inherited defect in the NF1 gene, and thus has formed multiple neurofibromas. Once activated, Ras can then bind to and activate MAP kinase, or RAF. Activated RAF can then phosphorylate and activate MEK, also known as MAP kinase. This activates MEK, and it will phosphorylate another kinase called ERK, which is also known as MAP kinase. Activated ERK can then phosphorylate substrate proteins and affect their activity, as well as phosphorylating transcription factors to affect or modulate gene expression in the cell. The MAP kinase signaling cascade is a classic example of signal amplification. For example, if each molecule of RAF is capable of phosphorylating and activating 10 MEK molecules, and each MEK molecule is capable of phosphorylating and activating 10 ERK molecules, you have an amplification of over 100-fold from a single activated RAS. Epidermal growth factor receptors are a family of 4 receptor tyrosine kinases called HER1, HER2, HER3, and HER4, HER being human epidermal growth factor receptor. This family is also known as the ERBB family. HER receptors function by forming homo and heterodimers with each other and transmitting mitogenic signals through the Ras MAP kinase cascade to promote cell proliferation. The HER1 receptor is commonly overexpressed in some forms of cancers. Overexpression of the HER1 receptor makes those cells sensitive to mitogenic signals or epidermal growth factor levels, which would, under normal expression levels, not be sufficient to stimulate cell proliferation. Truncation of the extracellular ligand binding domain of HER1 is also fairly common in some forms of cancer. In this instance, the truncated HER1 protein can dimerize and activate cell proliferation pathways in the absence of ligand binding. A monoclonal antibody called Cetuximab has been developed to target overexpression of the HER1 receptor in cancers. Binding of this monoclonal antibody to the extracellular domain of HER1 receptor prevents EGF from binding and stimulating cell proliferation. In some cases, binding of the monoclonal antibody will induce cell death pathways that will cause the cancer cells to undergo apoptosis. Some forms of breast cancer overexpress the HER2 receptor. The HER2 receptor has no known natural ligand. However, overexpressing HER2 induces ligand-independent dimerization with other HER receptors, causing ligand-independent activation of proliferation pathways. Trastuzumab, or Herceptin, is a monoclonal antibody that targets the HER2 receptor and is approved for treating HER2-positive breast cancers. Almost all receptor tyrosine kinases have a single plasma membrane spanning domain and are activated by ligand-induced dimerization mechanisms. The insulin receptor is also a receptor tyrosine kinase. However, it has a slightly different structure than most other receptor tyrosine kinases. The insulin receptor is a heterotetramer composed of 2 extracellular alpha subunits which bind insulin and 2 beta subunits, each spanning the membrane once and each having a cytosolic tyrosine kinase domain. The insulin receptor is already dimerized, even in the absence of insulin binding. Insulin binding to the alpha subunits, which activates autophosphorylation of tyrosine residues in the beta subunits. The resulting phosphotyrosines are binding sites for a family of proteins known as insulin receptor substrate proteins. IRS proteins have a phosphotyrosine binding domain, which allows them to dock to the phosphotyrosines of the autophosphorylated insulin receptor. The insulin receptor substrate proteins are then phosphorylated by the activated insulin receptor tyrosine kinase. The phosphotyrosines on the insulin receptor substrate protein are now docking sites for adapter proteins that link insulin receptor activation to multiple signal cascades. The insulin receptor substrate proteins can couple to the Ras MAP kinase cascade to stimulate cellular proliferation, to a phospholipase C pathway to generate second messengers like diacylglycerol and IP3, or to the PI3 kinase AKT pathway. Phosphoinositide 3 kinase, or PI3 kinase, is a lipid kinase that phosphorylates the 3' position of the inositol head group of phosphatidylinositide 4,5-bisphosphate, commonly called PIP2, to generate phosphatidylinositide 3,4,5-trisphosphate, or PIP3. PIP3 is a binding site for proteins with plextrin homology domains, or pH domains. Plextrin homology domain containing kinases are recruited then to the PIP3 on the inner surface of the plasma membrane. Among these is a kinase known as protein kinase B, PKB. PKB is also known as AKT. Binding of PKB to the PIP3 brings it into close proximity to PDK1, a kinase which will phosphorylate and activate PKB. Activated PKB can dissociate from the plasma membrane and phosphorylate substrate proteins, altering their activities to help insulin produce effects on glucose uptake, glycogen synthesis, and cell survival. The receptors for the transforming growth factor beta superfamily of signaling molecules have a similar mechanism as receptor tyrosine kinases, except they have serine-threonine kinase activity. They will phosphorylate substrate proteins on serine and threonine residues instead of tyrosine residues. All members of the TGF-beta superfamily, including activin and the bone morphogenic proteins, bind to serine-threonine kinase receptors, of which there are two types. Type 1 and Type 2 receptors are single-pass transmembrane proteins. Each has a serine-threonine kinase domain. The Type 2 receptor serine-threonine kinase activity is always on, phosphorylating itself in the absence of ligand. Binding of TGF-beta, or a bone morphogenic protein, to the Type 2 receptor induces dimerization with a Type 1 receptor. The Type 2 receptor phosphorylates and activates the Type 1 receptor. This mechanism is analogous to the ligand-induced autophosphorylation mechanism of receptor tyrosine kinases. The activated Type 1 receptors then recruit and phosphorylate a group of proteins known as receptor-regulated SMADs, or R-SMADs. Two phosphorylated regulatory SMADs form a complex with a co-regulator, or co-SMAD. This complex can enter the nucleus, where it binds to the promoters of genes that are regulated by TGF-beta or bone morphogenic proteins, and modifies their expression. Most cytokines signal through cell surface receptors that lack an intrinsic kinase activity, but upon binding of the cytokine, they recruit an intracellular tyrosine kinase and activate it, which can then phosphorylate downstream substrate proteins to produce the cellular response. All the cytokines, such as the interleukins and the hematopoietic growth factors, erythropoietin, EPO, granulocyte colony stimulating factor, granulocyte monocyte colony stimulating factor, as well as growth hormone and prolactin, all signal via these non-catalytic receptors that recruit and activate a cytosolic tyrosine kinase. Interferon gamma receptor, like the receptors for all other cytokines, signal via a non-catalytic receptor which upon ligand binding associates with and activates a cytosolic tyrosine kinase. Ligand binding induces receptor dimerization. Dimerization of the receptor allows association with a cytosolic tyrosine kinase of the family known as JAK kinases. JAK kinases were originally termed just another kinase, but have been renamed the Janus family of tyrosine kinases. Recruitment of the inactive cytosolic JAK kinase to the cytoplasmic domain of a liganded cytokine receptor activates the tyrosine kinase activity of the JAK kinase. The JAK kinase can then phosphorylate tyrosine residues on substrate proteins. Principal among these are the signal transducers and activators of transcription proteins, or the STAT proteins. Phosphorylation of a STAT monomer on a tyrosine residue by the activated JAK kinases creates docking sites for SH2 domains on other STAT transcription factors, allowing the STAT monomers to bind to one another to form a dimer complex that can translocate into the nucleus and bind to promoter elements present in genes that are regulated by the cytokine. There are two peptide families that bind to cell surface receptors that have a cytosolic guanylate cyclase activities. Both the atrial natriuretic factors and the guanylin factors bind to cell surface receptors that produce cyclic GMP. A and F factors are produced by the heart and bind to receptors in the kidney to regulate sodium excretion and therefore fluid volume of the blood. Guanylin peptides are released by goblet cells in the intestine and regulate fluid absorption in the intestine. In both cases, activation of the guanylate cyclase activity of the receptor increases cellular concentration of the second messenger cyclic GMP. Cyclic GMP activates another serine-threonine kinase called protein kinase G. This is very similar to protein kinase A activation by cyclic AMP. Protein kinase G phosphorylates and activates the cystic fibrosis transmembrane regulator, CFTR, altering chloride ion and fluid transport across epithelial cell membranes. Some forms of E. coli can produce a heat-stable peptide endotoxin, which binds to and activates the guanylin receptor. The activation of the guanylin receptor in the intestine results in overstimulation of the CFTR, which decreases water absorption in the intestine, leading to secretory diarrhea, explaining why one doesn't want to drink tap water while on vacation in developing countries. In the unliganded state, receptors with guanylate cyclase activity have a cytosolic domain which is heavily phosphorylated. Activation of the receptor by peptide hormone binding activates the guanylate cyclase activity, and cyclic GMP accumulates, which activates protein kinase G. One of the substrates of protein kinase G is a phosphatase. Phosphorylation of the phosphatase activates it, and it will dephosphorylate the cytosolic domain of the guanylate cyclase receptor. This inactivates the guanylate cyclase activity of the receptor, and the production of cyclic GMP stops, even in the presence of continued binding of the receptor by its ligand. This is a desensitization mechanism by which the cellular response to hormone binding is limited to prevent hyperstimulation of the system. Throughout the learning modules on cell signaling, I have referred to individual signaling pathways that appear to be quite linear. However, cells need to respond to dozens if not hundreds of extracellular signals. These signals have to be integrated to produce a coordinated response to the extracellular environment. Often, different signal transduction pathways will converge onto the same downstream effector proteins. This crosstalk between different signaling pathways allows the cellular response to be fine-tuned to the exact cellular stimuli being received. This diagram just shows a hypothetical convergence of hormone signals acting through different receptor classes to produce an integration of the cellular response mechanisms. A number of mechanisms are used to terminate cell signaling. The target cell may express enzymes that degrade the signaling molecule. For example, at neuromuscular junctions, muscle cells secrete acetylcholinesterases into the synaptic cleft to degrade acetylcholine neurotransmitters to terminate signaling. Acetylcholinesterase inhibitors then are used to prolong the amount of time acetylcholine remains active in the synaptic cleft to reduce the symptoms of myasthenia gravis, an autoimmune disorder which destroys the receptors for acetylcholine. Another mechanism to terminate cell signaling is activation of mechanisms to degrade second messengers generated in response to the receptor activation, such as the phosphodiesterases, which degrade cyclic AMP and cyclic GMP, or phosphatases can reverse the effects of activated protein kinases. Target cells may internalize the receptors to reduce their number at the cell surface and limit further signaling, or the receptors may be inactivated by a feedback regulatory mechanism that prevents the receptor from propagating a signal until the receptor has been reset to some basal state. Receptor desensitization mechanisms often involve feedback mechanisms that inactivate the receptor at the cell surface. For example, some G-protein coupled receptors are inactivated by phosphorylation of a C-terminal domain. This is accomplished by both cyclic AMP activated protein kinase A and also specific kinases called G-protein coupled receptor kinases, or GERKs. GERKs phosphorylate G-protein coupled receptors when they are occupied by a ligand. The unoccupied receptors are not substrates for the GERKs. GERKs act to feedback desensitize the G-protein coupled receptors. Eventually, cyclic AMP levels in the cell are reduced by the activity of cyclic AMP phosphodiesterase, and depending upon the receptor, the desensitization effect can either be rapidly or slowly reversed by phosphatases, which dephosphorylate the C-terminal domain of the G-protein coupled receptor, resetting it to the basal activity state. Here are a few questions to test your knowledge of cell signaling mechanisms involving receptor tyrosine kinases.